speaker is Dr. Jesse Goldberg, Associate Professor in the Department of Neurobiology and Behavior at Cornell University. He's going to be talking about neural circuit control of songbird behavior. And so, Jesse, you have 20 minutes, and you can go ahead and share your screen now and get started. Thanks.
Okay. Can people see my screen? I've already shared it.
Yes.
Yeah, we're good. Okay, great. Well, thank you for the organizers. It's such an honor to be in this conference, and particularly in this session with Now and Wolfram. In the spirit of Wolfram's upcoming talk, it's both rewarding and moving to have so many people here in the audience. So, you know, we're interested in neural mechanisms of motor learning generally, not just dopamine. And one reason why I think motor learning is so fascinating is because we come from very humble beginnings, especially humans. Human infants have a very reduced repertoire of movements. If you look at this video of a child learning to reach and grasp, you can see that the hand movements are very jerky. Even something as simple as grabbing an object is extremely difficult. And yet within about a year or two, this kid can be climbing trees, speaking argumentatively, and playing sports. So how is it that we gradually improve our motor performance with experience? Now, one of the ways that behavioral neuroscience has attacked this question is really within the traditions of B.F. Skinner and behaviorism, which is you can leverage the fact that thirsty or hungry animals can learn to do arbitrary motor skills for reward. And of course, many in this audience will be familiar with the experiments of Olds and Milner back in the 50s, when they showed that rats will not just learn to press levers for food, but also just to get parts of the brain stimulated that include the mesostriatal dopamine system. And as now introduced reward prediction area very clearly, we are familiar with this slide of recordings from midbrain dopamine neurons in a variety of species that show that they can encode a very specific type of reinforcement signal, reward prediction error, the difference between how good you expect something, how good the outcome was, and how good you expect it to be. And this happens when a thirsty monkey, for example, gets unexpected juice reward. You get this peak in dopamine firing. And a lot of theoretical work over the last decades has incorporated these key reinforcement dopamine signals, these phasic increases or decreases that now discussed, into a network architecture that can learn, for example, to link a stimulus or a state representation, for example, in the cortex converging to the dorsal striatum, into a reward maximizing action or policy. So for example, you can learn to move the joystick to the left when you hear a high tone, or move the joystick to the right when you hear a low tone. And we also know that the ventral striatum can compute the predicted value of states that now touched on. And so this is like a standard actor-critic architecture that the core of which is really this dopaminergic reinforcement signal. Now, this is all well and good when we take the assumption that animals are going around looking for rewards, which is the case with foraging. But most of interesting human behaviors are not specifically driven by specific stimuli or cues or states, and nor are they really in pursuit of explicit rewards. So if you take just a simple paradigm like learning to talk, let's consider the case of vocal babbling. Now, babbling is very interesting because we know from cerebral palsy and various diseases that affect kids that dopamine basal ganglia circuits are important for speech acquisition. And yet it's hard to think about how that actor-critic architecture and dopaminergic reinforcement signals could apply to a behavior like this. So here's why. Just consider what this kid is trying to do. So some aspects of reinforcement learning seem to fall apart here. First, there's no specific state representation or stimulus that's sort of dictating how he should move his tongue or lips or expiratory muscles of his respiratory system to make sound. So we don't really have a clear state representation, which you need for reinforcement learning. We also don't have explicit rewarding feedback. There's no juice reward for staying fast car, for example. And, you know, he's all alone right now. I'm secretly videotaping him, and he's listening to his own babbling. And so this doesn't seem like he's surprised or disappointed by any outcomes here. He's just babbling. But yet it involves the same circuits that we know would do reinforcement learning in a foraging context. So let me just play a game with you to sort of push the idea as a thought experiment of what a dopamine signal could look like in a situation like this if we were to impose on it an RPE-like framework. So let's just pretend he's trying to say a really fast car, which isn't so outlandish because at this age, if you say to him, point to the fast car, he will point to the Lamborghini, not the pickup truck. So his auditory system can parse language and form representations of sounds on the sensory side, so auditory representations. To say he really wants to make the sound a really fast car, but he doesn't yet know how to transform that auditory representation into vocal motor coordinates to move his mouth and tongue. So let's say on this next trial, he says a really fast far. Okay, so here we have, well, that's a mistake, right? He wanted to make a k sound, he made a f sound. So you could imagine that that's an error, all right? But if you really wanted to understand what might be the quality of this trial, a really fast far, you wouldn't just be able to look at the target of what he's trying to say. You'd have to look at the past history of his performance. So let's say, for all we know, he's never actually made the L sound before. The L sound is very difficult. You've got to get your tongue right up to the roof of your mouth, in the front, behind your front teeth. And many kids, when they want to make the L sound, for months make the W sound, a really fast car. So let's say on this particular trial, this was the first time he ever made the L sound. He just made a lucky guess and his tongue flicked up in position. Well, then it's conceivable that he's closer to the target than he thought he was going to be based on his past performance history. Now, under these conditions, you could imagine that this is a positive prediction error, that there's something better than expected that happened here, and that you could have a dopamine signal that would do something like this. It would exhibit phasic increases when your performance is closer to your target than you expected based on your past practice, and a phasic decrease when your performance is farther away than you were expecting based on your past practice. Now mentioned in his talk the idea that the dopamine system can do moment-by-moment evaluation of the state value or the reward. Here, it's an elaboration of that, where it's not just a moment-by-moment evaluation of reward outcome, but really a moment-by-moment evaluation of the extent to which your performance is matching the ongoing changing goals. Now, the question is, can the dopamine system do something like this? And I think, at stake in this question is to what extent, most of our daily lives are not in pursuit of rewards. So if you're typing and you make a typo, does your dopamine neuron have a pause, right? So I'm trying to motivate that what's important here is to the extent to which we can take our ideas of dopamine signaling and bring it to the real world in some sense, in the real world of humans, where we're mostly not seeking external rewards in our daily lives. Okay, so I'm going to briefly talk about how the Songbirds provide an opportunity to test this question, talk about dopamine signaling in singing birds and how that might be changed when females appear in courtship contexts. And then if there's time, I'm going to touch on this idea of whether or not there are multiple ascending RPE channels. Okay. So birds learn to sing. In zebra finches, which is the common lab animal, the adult bird sings a song that basically is about a second long. It's a sequence of syllables. And their muscles are super fast. They've got 7 muscles in each syrinx on each side. So there's 14 muscles total. The muscles can contract and relax about every 5 or 10 milliseconds. So you can imagine the adult bird song as being a 100 time step piano piece on a 14 key piano. And what the adult bird has to do is execute that piece perfectly in order to attract mates. All right. Now, baby birds, when they start to sing, it's like they bang randomly on the piano. They produce vocal babbling. Their syllables are widely distributed in duration, and they make all sorts of funny sounds. It's basically like babbling. It doesn't sound at all like an adult song. And gradually, over the course of about a month and a half, a practicing bird will converge and produce the song that he heard earlier in life, even if that bird did all that practicing all alone in a cage. So there's this idea that the bird is vocalizing. He's evaluating his own song based on auditory feedback and gradually getting better with practice. We know from classic studies of Konishi and Marler that if you deafen birds, they don't learn to sing. So auditory feedback is essential. As birds learn, the variability is reduced and the similarity to tutor increases. And it's been proposed that reinforcement learning may play a role in this process. Now, the key prediction of reinforcement learning algorithm over others like unsupervised or supervised learning signals is that there's an evaluation signal. So basically the idea is that when the bird is practicing, if he hits some note that's closer to the tutor than he was maybe expecting, then there'd be a better than expected outcome that might be associated with a dopamine burst. And on the flip side, you could imagine the bird could make some mistakes in which it has a worse than expected outcome, which would be associated with dopamine pauses. So we really wanted to test this idea of whether or not dopamine can evaluate this type of behavior. I'll go quickly through this, but birds are a nice model system because they have a discrete vocal motor circuit called the song system. If you take a slice of the bird brain, you can just see these nuclei jump out at you. This is what made Fernando Nottebohm famous in the 1970s, was the identification of the song system in songbirds. And about 20 years ago, it was realized that the song system is really not that different than, it's embedded in a four-brain architecture that contains a mesostriatal dopamine projection. So there's a VTA that projects to a nucleus called area X, which is like a striatopalatal structure within the song system. So this architecture, this dopamine projection to the basal ganglia, is very similar to what we know is important for reward learning in mammals. And to test if this projection could carry signals associated with the evaluation of birdsong, several years ago, Vikram Gadekar, a postdoc in my lab, recorded dopamine neurons in VTA, and we wanted specifically to record from those neurons that were going to this song system nucleus area X. So we used antidromic identification to record from these VTA X neurons that are known from past work from the Perkel lab and others, and Sam Sober's lab, to be dopaminergic. Okay, and then we had to control the perceived quality of song. And so to do this, what we did is we took a little snippet of syllable C here, and we would play it on top of syllable B. And we would do that probabilistically. So the bird is singing A, B, C, D, A, B, C, D, and sometimes you hear it's H, C, D, A, B, C, D, H, C, D. So there's these little 50 millisecond snippets of sound that are not aversive, but yet constitute a performance error. And we know from past work from Michael Brainerd's lab and Michael Fee's lab that these distortion sounds, birds could learn to change the way they sing the pitch of a target syllable, for example, to avoid the distortion. So we knew that the distortions were negatively reinforcing the song. And this is what it sounds like. You're going to hear slowed down song, and the first rendition is going to be undistorted, and you'll hear a little sound on the syllable B on the second rendition. So it's pretty subtle. I had to slow it down for you to hear it, but in the reality, the birds really care about this sound because they'll learn to change the way they sing to avoid it. Okay, so here's an example of an antidromically identified area X projecting VTA dopamine neuron, and the bird is singing. And here in red, can you see my cursor? Ben, can you nod if you see it? Or Ali? Yep, okay. So in red here is the distortion sound, and I'm just showing you shading when it happens. And you notice there's a little pause after the distortion. And in the spike raster, you can see this pause is extremely reliable when this target syllable is distorted. And you can see the phasic suppression in the rate histogram. Now that was the distorted renditions, and half the time we did not distort. So what happened then? Here's just the bird singing. Here's the spiking of the dopamine neuron. You see there's a tiny little burst here at the precise moment of the song when we did nothing, but when the bird might have been expecting an error. Because I should say that the birds have been exposed to this feedback for days before we do our recordings. And what you see in this raster plot is that these bursts after the undistorted time in the song were highly reliable. And you see a little peak in the rate histogram associated with the, what we interpret as the better than predicted song outcome, okay? And here's just another example of another neuron that had a particularly large positive prediction error. So the old idea is that the bird is singing and he's comparing his song to a tutor that he heard early in life. Here, we see that that can't explain both of these signals because of course it sounds worse farther from the tutor if you get distorted. But the question is, why is the burst happening right at the time of the song when we had probabilistically been distorting? And we wondered, maybe we've been training the bird to think that his target syllable sucks, right? So like not only is he learning to reinforce variations that eliminate that don't get distorted, but maybe he's learning that this is a part of the song, this syllable has a reduced quality. So to test this idea, we wanted to see if this is really a prediction error signal. And to do that, we had to control the probabilities of distortion. So here's a different experiment where we have a song, A, B, C, D, and syllable B, we're gonna distort half the time, okay? And an independent roll of the dice is gonna allow us to distort syllable D 20% of the time. So if we do this for days, and the bird in principle can learn that the predicted error of syllable B is higher than the predicted error of syllable D. So basically the bird can learn like, you know, I got my song A, B, C, D, My syllable B really sucks. My syllable D sucks a little bit less, and my other syllable A and C are fine, right? And so if the bird is really learning the predicted value or the predicted error of each syllable, then that should be evident in the dopamine signal on the undistorted renditions. So if we look at the undistorted renditions, the prediction error would be larger following the undistorted rendition of the high probability target syllable B. Okay, does that make sense? And so that's exactly what we found. When we recorded these VTA-X neurons, we found that the bursts following the undistorted rendition of the high probability target were always larger than the ones in the low probability target. To make this easy to understand, I play tennis. My backhand sucks, my forehand's good. So if I hit my backhand in, that's more surprisingly good to me. So my dopamine signal will be bigger following a backhand winner than following a forehand winner, even though they both get me the point, okay? And so in this metaphor, target 1 is like the bird's backhand, and this target 2 would be like the forehand, okay? So what this tells us is that what the bird is doing is comparing its ongoing performance, not just to a fixed target, but rather according to flexible benchmarks that can change with practice. Okay, and I'm not gonna go into details here, but we had a paper in Neuron last year, and I also encourage you to look at papers recently from the Mooney lab or Michael Fee's lab that put this dopamine signal in a larger context of auditory inputs to the dopamine system that can drive the error, of ventral, palatal, and ventral basal ganglia inputs that can compute the predicted syllable quality, and dorsal striatal circuits in area X that can use this dopamine signal to learn a reward maximizing policy. And so really, I think what we have when you zoom out is that the songbird has the same architecture as the mammal in the sense of you got ventral striatal circuits for computing predicted reward value, dopamine signals that compute RPE, dorsal striatal circuits that can use that RPE signal to learn a reward maximizing policy, that this might be an ancient architecture that can be repurposed from foraging for something like vocal learning. Okay, now I'm going to pivot here to what this behavior is really all about for the bird. They're trying to show off their fitness, and they're trying to mate. And all of the data I just showed you were from birds singing alone, practicing. So if you put a bird alone in the cage, he just sings around. Now, when the female appears, it's a dramatic event for the male, especially if he's been alone in a box for a week, okay? The female appears, and this is a video that Mike Long shared with me. I really love, we have videos like this, but they're not as good as this one. Watch what this male does when the female appears and when she disappears. Okay, so based on that video, you can see this is female-directed song. Female-directed song, the males are actively in pursuit of the female. If it's in a tree, you'll see that the male will follow the female around and sing at her vigorously. And there was evidence that the VTA to area X projection had something to do with this female-directed performance. So for example, the trial-by-trial variability in song is dramatically reduced when the male sings for the female. And some pharmacology and microdialysis experiments had hinted that maybe it's an increase of tonic dopamine levels in area X that are mediating the ability to reduce the variability when the female is present. And so we thought there was an opportunity to test ideas about phasic and tonic dopamine signaling in the system, because what I've showed you so far is that when the male's alone, he's singing, he's got these error signals. Now what happens when the female appears? Does the spiking change? Does the baseline firing rate change? Do the error signals go up or down? And so this is a very clear-cut experiment with very easy to interpret outcomes to see what happens when the female appears. It's a clear state change.
Hey, Jesse, I just want to pop in to give you a 2-minute warning.
Perfect. Okay. So here's a neuron, a male singing alone. We saw the error signals again. The male's practicing alone. We observed these error signals again. Now we can record the same neuron. So we just introduced the female, and we can ask of a given neuron what it does. And so the first thing we observed is that in most of the neurons, the error signal went away when the female was there. It was a little bit left in one neuron, which is what's giving you this on the average. But the error signals were dramatically reduced during female-directed song, because we still kept the distortions going. And this gave us the idea that maybe the male is not so much attending to his own song. He's not evaluating himself. And maybe since he's focusing on the female, maybe he's focusing on what she might be doing. And consistent with that idea, what we saw is that when female made these brief affiliative calls, this is now the female call, these same VTA exons could exhibit these phasic bursts aligned to the female call. And so this is basically these dopamine neurons can change their tuning depending on the courtship context. When they're alone, they're evaluating, it's a self-evaluation performance error signal for learning. When the female appears, that signal is dramatically reduced or in some cases gated off. And those same neurons can now be activated by aspects of the female behavior. In this case, it was just a vocalization, but this is really the tip of the iceberg for what dopamine signals can be doing during these courtship social interactions. Okay, I just want to end with one last point, and it won't be long. If you think about it for an instant, reinforcement learning signals for vocalizations are extremely problematic because if this bird sung a beautiful song, better than expected, right next to a snake's nest, you would not want the bird to learn to prefer that place, right? Animals vocalize as they move around. So it would make sense that they would have different reinforcement streams for vocal performance evaluation versus place evaluation. And so we tested this idea with perch contingent sounds and lights and vocalization contingent sounds and lights. And I'll just cut to the chase, which is that the birds could learn to avoid a perch that was associated with strobe lights, which are aversive. But the bird could not learn to change the way it sang its song to avoid the strobe lights. So there was the birds in the classic Marty Seligman psychology literature, it'd be the contra-preparedness. The birds are contra-prepared to leverage strobe light signals and access those to the vocal system. And similarly, the birds preferred to occupy perches that were associated with these brief noise bursts, even if they wanted to change the way they sang their song to avoid the noise bursts. So the punchline from these studies, which we published if you're looking at interested in a couple years ago, is that a global reinforcement system can't work to explain what the bird was doing in these experiments. They would need to have reinforcement signals that are specifically dedicated to analysis of the song and route that to the song system. And you've got a lot of other dopamine neurons that could be doing other things, such as evaluating the value of a place. And here it's worth pointing out that in these experiments we recorded from VTA, it was a tiny fraction of the neurons in VTA that encoded these error signals that I talked to you about, but those were the ones that were anadromically identified as going to area X. So I want to introduce the idea that there can be multiple dopamine channels that can evaluate different aspects of behavior beyond reward. Thank you very much for your time. I guess I'll just leave you with the summary is that you can common architectures and signals can underlie reward and performance learning. Dopamine signals can be gated by social interactions and context. And I want to thank Vikram Gadekar, the postdoc who did these recordings, who's starting his lab at Columbia this year. Raydong Chen, a grad student, just finishing up in the lab, headed to MIT. Pavel, a postdoc, Andre Roser, a grad student, and Alex and Eliza, who did these dopamine recordings. And thank you very much. And hopefully there'll be time for a couple questions.
Okay, great. Thanks, Jesse. I'll just sort of start with a sort of broad question. The sort of notion of context dependency, I think, is really interesting. And have you done much work or thought much about the system working in a context where males are singing with other males that aren't tutors in maybe a more competition-related setting and that kind of thing?
So yeah, I mean, there's a lot of behavioral literature on birdsong, and males do sing directed song to other males in a kind of, not necessarily adversarial, but in a hierarchical setting where they're trying to jockey for position or pecking order. That really is the case. And we haven't done those experiments to record dopamine signals under those conditions, but my guess would be it'd be the same as in the female, in that they're gated off because the song is quantifiably different when it's directed, and males will sing directed song to females, to children, or to juveniles, and to other males. So I think that this alone context is the practice context in which you're going to get these self-evaluation signals.
Cool. Okay, well, I'll hand it over to the co-mod, and there's a few questions from the audience.
Okay, hi Jesse. First question from Alexa Diambra, and she asked whether these manipulations were done in adolescent birds or adult birds, and more specifically, whether these feedback error signals will change depending on what stage of learning is the bird in?
Yeah, that's a great question. We did these experiments in young adults, mostly for methodological reasons. We would love to do these experiments in younger animals and babbling birds. We haven't done that yet. I think that's something Vikram might take up in his lab. I should say that at the very early onset of vocal babbling, I think it's highly unlikely that the dopamine system is at play. Because in order for these signals to exist, there have to be predictions about the outcomes. And when you first start banging on a piano randomly, you're not necessarily even having goals. You're just figuring out what happens when I press this key, what happens when I press this key. And in machine learning context, there's often periods of supervised or unsupervised learning that occurs before RL frameworks start to play. So it's conceivable to me that in the early stages of babbling, the birds are just learning paired inverse and forward models for what happens when you make different sounds. Then they start to have an infrastructure, maybe in loops between cortical, auditory and motor cortical areas that involve the predicted consequences of sounds. And then you have to impose value on it relative to the tutor. So I don't necessarily think that the RL story I just told captures everything going on in birdsong. This is, I think, what happens maybe in the later stages of learning when you've already got internal models for vocal control.
I would like to actually follow up on that and combine that with a question from Lindsay Cameron. The question is, how would these error signals change if these animals were raised in isolations? Would they have a problem inferring the correct feedback for example, a courtship context?
Yeah, so that's a really great question. I mean, isolate birds never really crystallize to a fixed song, but they do develop some stereotyped syllables. So it's unclear what their internal model is. It's a really deep question, actually. I would guess they have some basic template. They got to be trying to do something, but that's just an assumption that could be tested. You could repeat these experiment in birds that never got a tutor and that were singing funky isolate bird songs, and then test if these signals would still exist. That's a good idea.
Excellent. I have another question from Ryan Kowaleski. This question is about the instantiation. You talked about different channels at the end of your talk, and he asked whether these channels are in different underlying anatomical connections, maybe the same set of connections gated with a context-dependent activity. How would you think these different channels could be instantiated in the circuitry?
Yeah, so this is really, I think this is a major open question in the dopamine field generally, this idea of channels. We're all working with reward and punishment. Animals are out there doing a lot of other things, right? Like when they groom themselves, they probably like to have a certain sensation on their tongue, right? What's evaluating that? So in the song system, the VTA neurons that go to area X are not in a nucleus. They're just a random smear of scattered neurons intermingled with other neurons that are not going to area X. So it's not just like a medial lateral ventral dorsal thing. I mean, yes, they tend to be clustered more anterior VTA, but there are neurons right next to VTA X neurons that are going somewhere else and signaling something different. We know that because we can sometimes get multiple neurons on an electrode. I think that there are yet to be discovered learning rules that configure who wires up with dopamine neurons and what those dopamine neurons wire back to. And I think that that's related to the fact that, as now showed in a neuron paper with whole brain imaging lots of different inputs of dopamine neurons, these inputs can basically signal every conceivable reward-related and prediction-related signal, both positive and negative, and yet somehow dopamine neurons will read that out and create a pretty homogeneous RPE signal in a Pavlovian context. So there's got to be some learning rules that, even in the fetus, that will say, like, you know, dopamine pause and burst dependent LTD and LTP on inputs that will wire up almost in the way that retinal waves wire up the visual system, I think are wiring up the dopamine system and its inputs so that the system is learning to not generate dopamine pauses or bursts when nothing's happening under spontaneous conditions. That's just pure speculation, but basically during development, before an animal even is in the world, just in a fetus, you'd want to have a way to make sure bursts and pauses don't spuriously happen spontaneously. So there's got to be some, I imagine that you'd have some optimization principles to train that up, but that's just pure speculation.
Okay, this is just a personal follow-up. Do you think there could be experiments to test what you just asked about dopamine activity in the fetus, for example?
Yeah, I think you could do some with optical and genetic methods, like do some manipulations with dopamine inputs in utero and like show that the wiring gets all messed up. I mean, we actually have an experiment we're doing in the lab right now to see if deafening might totally distort the specificity of the VTA X projection. So in birds, like VTA neurons go to area X, and then they also go to this ventral palatal region, but not much other places, like the part of the strand that might be involved with like nest building. I don't know. So it's like we have a kind of a channel in the bird. But I suspect that's all wired up in an activity-dependent way. So by messing with the activity, we might be able to like mess with the topography of at the anatomical level. Very easy experiment, actually. But that doesn't get at the deeper question of like what happens in the fetus.
All right, thank you very much. I think we're out of time for questions. These
Thank you
questions,
all.
the remainder will be posed in the Slack channel.